we had a garage with a driveway, and there was a huge anthill that got destroyed by my parents every year. But I loved when the ants would come back and they would build their hills and do their things. And I would get out a bunch of different kinds of cereal from the house, sugary and non-sugary, and lay it out and watch the ants figure out what they want to do.
Experiment.
It was experimenting to see what the animals preferred, and then how did these ants actually take those giant pieces of food back to their hive.
From the heart of The Ohio State University on the Oval, this is Voices of Excellence from the College of Arts and Sciences, with your host, David Staley. Voices focuses on the innovative work of arts and sciences faculty and staff, with departments as wide-ranging as art, astronomy, chemistry and biochemistry, physics, emergent materials, and mathematics and languages, among many others. The college always has something exciting happening. Join us to find out what's new now.
I'm joined this morning by Jeanne Serb, professor and chair of the Department of Evolution, Ecology and Organismal Biology at the Ohio State University College of the Arts and Sciences. Her research examines how biological diversity originates and is maintained through the interactions of multiple levels of biological organizations. Dr. Serb, welcome to Voices.
Thank you so much.
And I want to start first with, you describe yourself as an evolutionary biologist who's interested in how organisms sense their environment.
Yes.
Why that question? What drew you to that question or that problem?
I think the most important thing is that when we consider organisms, oftentimes we kind of think of them in a vacuum, right? We're watching them do their behaviors, moving around, maybe interacting with another individual. But we don't think very often about what kinds of cues are they taking in? How do they know what's happening in their environment? How do they make the choices to move in a particular direction, to go into a particular habitat, to interact with a particular individual? And all of those things differ between the organism that you're thinking of, right? For humans and lots of vertebrates, we think of visual cues being the most important thing. Our sight drives most of our decisions, right? You can think of it in the case when you are looking at a commercial, right? How is the advertiser trying to attract your attention so that you buy a particular thing? How do you pick out the clothes you're going to wear? Those are oftentimes visual decisions. But you look at another type of organism, and that sensory system may be shifted from a visual focus to a chemical focus. And instead, they have a whole new world that we as humans don't necessarily see. We can't appreciate because we don't use scent in the same way or taste in the same way. The best way of thinking about that is if you ever walk your dog in the winter, right, and a fresh snow has happened, you get to see what your dog is smelling on that walk, right? Because you're walking through and there is, on that fresh snow, the traces of the last dog that walked by, and your dog is stopping and picking up that scent. Right? But you get to see it for the first time, even though you don't sense it in that same chemical way.
What drew you to this problem initially? Of all the things you could study as a biologist, why sense? Why sensing the environment?
I think the thing that grabbed me first was thinking about eyes, because eyes have evolved across all animal life probably 60 different times. So we get very narrow human focused, right? We think about the vertebrate eye, and we may think about how the human eye may differ from the eye of a fish. But that eye type, that camera type eye, is a single evolutionary origin. It happened once across all vertebrates, and it became specialized in humans, in the eagle that you may look at the deep sea fish you may look at, but it's all the same eye. If we back out of the human or vertebrate world, right, and think about how other organisms see, look at the lowly jellyfish, right? We don't think much about the jellyfish, yet there's a whole lineage of jellyfish that have 24 different kinds of eyes. And some of them are camera-like eyes like our own, where they have a lens and they have a retina, and others are more simple, where they're just being able to detect the direction of light. So thinking about how did this morphological, the structural diversity of eye types that we see, where did it come from? And then the most exciting thing in an evolutionary sense is, regardless of how the eye is structured, the genetic mechanism, how the eye actually senses light, the proteins that allow it to sense light, is exactly the same from jellyfish to humans. And so my interest is thinking about those different levels of how change has happened over time. You have this relatively conserved genetic level, and yet the change that's happening at the structural level of the organ of the eye is really, really different.
So let me go back. 24 different types of eyes.
Technically, 6 different types of eyes. There's like 4 of each because of the way they're arranged.
A single jellyfish.
In a single jellyfish.
So you have to explain. What explains that? Why that evolutionary development?
Yeah. So, I mean, this is the thing that becomes really exciting. Is like, why do these organisms that we think are very simple?
By the way, I didn't realize jellyfish had eyes. I want to go on record saying that.
Yeah, most people don't. And it's not all jellyfish. It's this one lineage of the Cubesonian jellyfish. And these animals have the jellyfish structure, the kind of the bell-shaped, little tiny short tentacles. And underneath the bell, there are 4 different structures that hang down called rapellium, and they have the different types of eyes attached to them. And we think that jellyfish are using visual cues, this particular jellyfish group, because they're moving through mangroves, and they need to be able to direct how they swim so they don't hit these mangrove roots as they're going through the water.
So I don't know how to ask this, I guess. Why do we have only one singular type of eye?
Yeah.
Why don't we have 24 or 4 or 6?
I mean, so this is what's exciting about evolution, because in order for you to understand how a particular lineage has changed over time, you have to think about what the ancestral condition was. And in vertebrates, we are highly conserved in our structure. We have pairs of everything, right? Ears, eyes, right? Our limbs, we're very bilaterally symmetric. And so there isn't the opportunity. We're constrained in some way in evolution to think about the body patterning. And so how we go through an embryonic development limits what we can have as far as different types of structures, in this case, the pair of eyes.
So most people think of eyes, as I was suggesting right there, as a single invention by evolution. You found they've evolved at least 5 separate times.
Yes, in a group of bivalves that I study. These are marine bivalves.
So take us through these 5 stages, or what this process was like.
So I work on a group of marine bivalves called the pteramorphians. These are the tasty ones. So they're the ones you want to eat.
So they sound like superheroes initially. No,
yeah. So they include the scallops, right? And you will ask, do scallops have eyes? Yes, they do. Tens to hundreds of eyes. And then oysters, which as adults don't have eyes, but their larvae have an eye spot, and we'll talk about that. And then mussels. Those are like the main groups that people are most familiar with. The first question that comes up when I bring this topic to a conversation, they say, I didn't know scallops have eyes. I've never seen them. I've eaten in my pasta all the time. And as it turns out, when you eat a scallop, you're basically eating the bicep of the animal. The body that lines the 2 shells is thrown away in North America. Most North Americans don't eat it. Other countries tend to eat more of the animal, but we just eat this muscle that holds the shell together. So in my research group, what we found is that when we built what we call a phylogeny, so this is a bifurcation illustration showing how organisms are related in a bifurcated way.
Meaning splitting
off like a tree. So we can think of it as a tree, like a family tree. And we then went through and looked at when these different eyes have arisen on that tree. We found that there were 5 different originations of eyes. So this goes from relatively simple pit eyes, where they're basically a kind of a pit or a pocket that forms in the tissue and is lined by a layer of pigment. And then it has like a retina. It has a bunch of photoreceptor cells like a retina in it. And that allows for direction of light to come in from multiple directions, and the animal to be able to tell what the direction of light is. It's relatively simple. It does not create an image, but it gives directionality, and then the animal can move towards or away from light. These eyes can become much more complicated. In the case of one of the other groups of the pteramorphians, which is these arc clams, they actually have compound eyes like you would see in a fly, like an insect. But they are evolved in a totally different way from a different origin than what we see in arthropods and insects and crustaceans. And then scallops, which is the group I focus on the most, they have the most complex eye. It is a single-chambered eye like our own. The light goes through a cornea like our own, and then there's a lens, and the lens directs the light to the back of the eye. And in a vertebrate eye, we would have our retina there. And a scallop has something totally different. They have a crystalline mirror that reflects the light back into the center of the eye, where they have 2 independent retina that are made of different types of photoreceptor cells. And what's really cool about the scallop eye is that they respond to light in 2 different ways, turning on and off, depending on the presence and absence of light. That first retina is allowing them to see basically structure in their environment on kind of the peripheral edge of their visual field. And then the distal retina at the top is allowing them to see movement. And so they see these shadows moving over their visual field. And these two retina work independently of one another. Each has their own optic nerve. And one of the unanswered questions is, how is this visual information processed by the organism? Because part of it happens in the retina, and then presumably part of it happens in a ganglion that they have, which is their smaller version of a brain. And they have big optic lobes there that are processing the information.
Is that a question we'll be able to answer?
I think so. There's another group, a colleague of mine, Dan Spicer, who's at the University of South Carolina, and his group have been working on some more behavioral assays to understand how the animal's processing that visual information. There's been some other work in the 1960s and 1930s looking at the physiological side of it, but I think this is an area that we are poised to address with a lot of new technical advances.
Are you able to describe what it means to see like a scallop? I mean, it's been said of other animals, I suppose.
Yeah.
Do you have that capacity?
That's a great question, because a lot of the processing that happens for vision happens within the brain, right? And that's a harder thing to understand in an organism where you can't do the same types of behavioral trials that you can do within mammals or even octopus, right? I think we can have a good idea based on the optics, right? How those photoreceptor cells are packed, and we can understand the level of resolution that an image might have. We could probably do some behavioral work with them to kind of have an idea of like what they prefer to move into for habitat. But yeah, it's like, how do you think like a scallop, right? And you can't really get into the ganglion there, right? Not the brain, but the ganglion.
So how did you settle upon, I guess, scallops? Why study scallops?
Yeah.
Of all the things you could
bivalves
study,
in the world,
yes.
right? So when I was working on my PhD at the University of Alabama, I was actually working on freshwater bivalves. So these are non-eyed, eyeless bivalves that are found in freshwater streams, rivers, and lakes throughout North America, throughout the world. As it turns out, their greatest diversity is in the southeastern United States. And so I did some work there. And then I graduated and wanted to move into a system where I could do some more experimental work as an evolutionary biologist. And that's hard, right? Because when we think about evolution, oftentimes we're thinking about big geological periods of time where change is happening. So how do you think about evolution in the context of a period of time that you can study? And there's things that you can do in bacterial systems, for example, and there's a few other things that you can do in some other populations of organisms. But for eye evolution, that became a little bit more of a tough question. And I was sort of moving into this idea of a system where I could manipulate. And it turns out eyes have, because of that conserved genetic property, all of the eyes have the same type of protein used for light sensing. I wanted to develop a system where I could look at how do you change the protein sequence and see how it changes how the organism can sense their environment. And so I ended up working with Shozo Yokoyama down at Emory University a good 10 years ago, maybe 15 years ago. And he had developed a system where you could express these proteins in cell culture outside of the animal. Wow. So what you can do is you can isolate the gene. We now have that as a relatively simple thing to do with some of our next generation sequencing approaches, right? You can determine what in a single tissue, all of the genes that are being expressed there at a particular time point. You can then use bioinformatic tools to identify your proteins of interest, which we can do. We know these things that we're interested in are called opsins, and they have very specific signatures in their motifs of how the sequences are arranged, so it's easy to pull those out. I can send them off to a company and have them synthetically made. I don't even have to isolate the genes myself. I can have this company do it. And then we place these genes into a circular piece of DNA called an expression vector and insert that into mammalian cells. I grow them in cell culture, and those mammalian cells are like little protein factories. They make the protein I'm interested in. I isolate that, and then I can use a UV spectrophotometer, which is a special machine that walks through basically nanometer by nanometer of light and watches this curve develop showing how that protein absorbs that particular wavelength of light. in a scallop eye, they have 18 different opsins they're expressing at one time. We can start looking at what types of light are they responding to, and can they see color? How are they seeing contrast in their environment? Do they see UV? All of those things we can start putting together.
So I'm going to make certain I understand this. So you mentioned opsins now
Yes.
a couple of times. This is, well, the same proteins, opsins, are found both in scallops' eyes and in
humans.
our eyes.
Yes.
What are the implications of that?
So what we're really interested in is understanding how that protein changes over time to allow an organism to sense different types of light, right? So in a human eye, we have 4 types of opsins. One is in our rod photoreceptor cells, and that allows us to see not color but light. And so especially in dim light environments, we use our rods. In bright light, we use 3 different cones, or most people, not everyone. Colorblind people are missing a cone. In most people, we have 3 cones that allow us to sample light in the red, green, and blue part of the spectrum. And even though we only have those 3 cones, the combination of signals coming from the excitatory response of those cones allow us to see all the different colors in the rainbow that we're able to see, right? What we can do by manipulating those different types of opsins is we can understand how does that shift happen going in from one environment to another. So if you are a bird, for example, you actually have an extra cone. You have an extra opsin that allows you to see in other parts of the visible or UV spectrum. Same thing with insects. It's a way that we can better understand how organisms adapt to different types of light environments. How do organisms maybe have secret signals? This happens a lot in communication between males and females, where a male may have a pattern that he wants a female to see, but he doesn't want a predator to see. And you can do that by hiding it in a particular color channel that a predator doesn't have access to, but others of your species do. And so it creates, again, this hidden communication that's happening that's very species specific, that's fascinating to me in that evolutionary context.
My understanding is you went into this research thinking that species with more eyes or more receptors would have more opsins.
Yes.
And that's not necessarily the case?
That's not necessarily the case.
You have to explain
Yeah.
this anomaly, or what does this throw away all your research now? Or?
But it redirects my research in some way. So what we did, it was during COVID, and everyone was locked out of their labs, right? And so we were trying to think, my postdoc, Kyle McElroy, and I were trying to think of ways that we could develop a project that would be based on public databases, right? DNA found in public databases that we wouldn't have to go into the lab to look at. What we basically did was go through and mine using bioinformatics tools to go through and look at a bunch of genomes that were available or transcriptomes that were available for 80 species of mollusks, going from cephalopods to gastropods to bivalves. What we found was that what we were looking for was there an association between complexity of the eye or presence of the eye and number of opsins there. And as it turns out, that's not the case. One of the most complex eyes that people are aware of in mollusks are cephalopods, and they have very few opsins. And then you look at something like zebra mussel, these little tiny invasive mussels that come into our waterways in North America, and these things don't have eyes at all, and yet they have like 36 different opsins. So the question is that when you're looking at a genome, it's basically a long list of all the genes that are in an organism's DNA, right? But it doesn't tell you when and where and why those genes are there. So in order to understand the context for those genes, you'd have to take a different approach. And we call this looking at gene expression. It's a type of sequencing of transcriptomics, which is basically the transcript of a particular gene in a cell at a given time at a given place. And so basically what we want to do is say, okay, there's all these opsins. There's no presence of eyes. These opsins have to be expressed somewhere. Where else might they be expressed? And bivalves are really cool, and this is true for a lot of marine organisms, because they go through a larval stage that looks very different than what you see in the adult. This larval stage are tiny, microscopic, maybe get to the size of a sesame seed, okay? And at this stage, these larvae are swimming. They're very mobile. They tend not to feed very much. They tend to eat a little bit, but this is their dispersal period. And they respond to different light. And so they move depending on the stage as they're going through the swimming stage. Depending on the time point, they may be attracted to light at some points and then not attracted to others. So there must be something in those larvae that allow them to sense light. And many of them have an eye spot, not an eye that can reconstruct the vision or the structure in its environment, but detecting light. And so our next question was, well, is this larval stage, is this where opsins are being expressed, and if so, why? And so we kind of got into looking at a gene expression approach, looking at these different larval stages. And what we found is as these larval stages approach a period of time for metamorphosis, when they go from the larval form to the adult form, just like you think of with the caterpillar and the butterfly, right. Many marine species do the exact same thing. And they have to find their preferred habitat, right? So the place that they want to stay, because they go from a swimming larvae to your typical bivalve that is attached or doesn't move very much as an adult. And so it becomes that decision of where to do their metamorphosis becomes very critical. It's a life and death decision, honestly. So having good sensory cues are going to be really important for that larva to know that this is the right place to settle for the rest of its life and build its home, essentially, right? And as it turns out, opsin expression is upregulated, seems to increase in the expression as it approaches that period of competency to become the adult form for that metamorphosis. So there's one other thing I want to say about opsin, because it's such an interesting gene. And I should say it's a superfamily. It's a bunch of different genes that are in this group. We know that most of them are light sensitive because they have certain signatures in their sequence that allow us to identify that. And we can test that in the lab. But we know from some of the model organisms, mouse being a good example of that, where we've done a lot of different kind of research, mostly biomedical research, right? That opsins sometimes aren't just used for sensing light. As it turns out, they're probably the Swiss Army knife of sensory systems, because what opsin can do is it also can be used for chemosense. It's been demonstrated to be used for temperature sensing. It has been shown to be expressed in ears of vertebrates. And so opsin probably has many different jobs that it does. For an evolutionary biologist, this is a gold mine because we know that most genes don't have one job. When we're taught about genetics in school, oftentimes we're given the most simple explanation, right? Because we want to understand the concept. But then when we look at how biology actually works, it's usually a much more complicated story. So opsin is nice because we know that you change the sequence of opsin, and now the organism can perceive a different wavelength of light. There seems to be a one-on-one interaction there. But if opsin is used in some of these other contexts, for example, sensing a chemical, then that means there's going to be part of the protein, part of that gene that's constrained to still sense light in some contexts, but also still needs to be able to sense a chemical, right? Unless it totally dissociates those two jobs and only does one really well. But if it has to do two different things, that means there's a pull and push trade-off that's happening. And that kind of constraint is really interesting when we think about how genes work. And we have a really good system to start playing with that trade-off.
As we've been talking about vision, and I'm not talking about your work here, but is there anybody that's been looking at vision and artificial intelligence? I know that Waymos, for instance, can, quote-unquote, see their environment because they have lots of cameras.
Yeah.
Are these analogous processes? And is anybody thinking about the implications of the biologist's work to talk about the vision of artificial intelligence?
That's a great question. I don't know if anyone's doing that. I can say for opsins, there's another interesting application that we aren't doing in my lab, but some of the things that we do may help inform directions that research may want to go, and that is optogenetics. Have you heard of the term optogenetics?
I don't think so.
This was one of the most important research directions that happened in 2010, I believe, is when it first happened and people started learning about it. It's basically a way where you take a light-sensitive protein, an opsin. They're usually mostly bacterial opsins, but I could see where other types of opsins could be used, like from a mollusk. And you express that protein in a cell in a living animal where it's not supposed to typically be. For example, the brain. And there's a great series of YouTube videos where they do this in rats. They express the opsin protein in the brain of the rat, and then they have the rat wear this little light hat. It's worth seeing. And what they are able to do is by having that opsin there, they can basically genetically dissect the different circuits of the brain to understand feeding, fear, aggression in the rat's brain. There's a couple of really great videos. One is of a rat that is obviously very well fed, shall we say. It has its little light hat on. It's not hungry. They turn on the light, and the rat immediately goes to the food and starts eating it. And they turn off the light, and it stops eating. And you can just do this back and forth, back and forth. They've done a similar thing with aggression, where another part of the brain, they express these opsin proteins so that basically it's overriding that circuitry. They turn the light on, and it is I think it's like a bottle cap in with the rat. It's nothing. It's innocuous. And yet, as soon as the aggression circuit is turned on by light, the rat attacks that object, and it's just being controlled by that circuit. What's really cool is that they're thinking about using optogenetics in ways to help control things like diabetes, schizophrenia. There's depression. There's a lot of different ways that humans may have this technology available to them in the future. And I'm really excited to see where that's going to go.
Well, since you mentioned technology, so you are fairly new to Ohio State. You're still in your first year here.
First 6 months.
First 6 months. You came from Iowa State, and among other things at Iowa State, you were director of the Office of Biotechnology.
Correct.
First of all, tell us what that office was. And do we have anything like that here at Ohio State?
The Office of Biotechnology at Iowa State was a centralized area where we had shared instrumentation that was used across all the university, but other academic institutions and private industry. It was a great way of partnering between what we could do at the university level and also with private industry. We oversaw about $18 million worth of instruments, from doing different types of DNA sequencing, microscopy, proteomics, metabolomics. We did it all, and it was based on fee-for-service. What was really exciting about it is it became a central resource then for this huge research community. And it also served the global community. We had people sending specimens or samples from all over the globe, including Asian countries, African countries, European countries. And we also worked with many other industry. So what it allowed us to do as that partnership is that we might identify a need from a series of researchers for a particular type of instrumentation, and we could partner with those people so that they didn't need to necessarily invest in their research and development team for an instrument that may only serve their need for a short period of time. We would collaborate and purchase instruments together. It would help us direct where we need to go for the next type of purchasing. Most importantly is that it wasn't just purchasing an instrument. We had people in our 9 facilities that had 20 plus years of experience working in that field. So you as a researcher could have a great question, but not know how to answer it. And we could sit you down at a table with 5 experts from DNA, proteomics, metabolomics. I'm not sure what I need to do. Here's my question. And we could sit down and brainstorm and come up with an experimental design that would get you to the answer that you needed. And so you didn't need to necessarily be trained in that or have experience. We provided all that work. And then you could just drop off a sample if you wanted to, and we would run the sample and give you the data. Or if you need to train personnel in your lab, we would bring them in and actually have them learn how to use the instrumentation. So it had a great potential. One of the things that I regret that I wasn't there long enough to do is that it would have been really great to have an undergraduate degree in biotechnology so that we were training the next generation of researchers and scientists to apply these methods into other parts of work.
And we have that here.
And we just got approval for a biotechnology major in our college, yes.
Mm-hmm. It seems to me Ohio State could be doing these same sorts of things, right?
Absolutely.
Even absent an Office of Biotechnology.
Yes. And so we have centrally located instrumentation, especially with the biomedical side of the university, and so that's going to be very useful. But oftentimes when you're working with something like biomedical or you're doing work with veterinary medicine, which is another place oftentimes you have this type of instrumentation, they work with one type of specimen, right? In biomedical field, it's mammals. You work with mice, and you work with human samples. What we were able to do at Iowa State, and what I hope that people think broadly about at Ohio State, is that the research interests are going to be way more diverse. It may be plant-based. It may be bacterial-based. It may be working on slimy organisms like I do with mollusks, right? And so each of those have their own challenges with handling those specimens, preparing them for the instrumentation that you might want to put that stuff on.
Why are you a biologist? Did you know when you were 4 years old that you were going to be a biologist?
I probably was about 6.
Really?
Yeah. I loved watching animals move through their environment and do the things that they do. My parents, unfortunately, were not very outdoorsy people at all. So I was limited to what was in my backyard. We had a garage with a driveway, and there was a huge anthill that got destroyed by my parents every year. But I loved when the ants would come back and they would build their hills and do their things. And I would get out a bunch of different kinds of cereal from the house, sugary and non-sugary, and lay it out and watch the ants figure out what they want to do.
Experiment.
It was experimenting to see what the animals preferred, and then how did these ants actually take those giant pieces of food back to their hive.
It strikes me that today, and correct me if I'm wrong, biology has become a data-centric science.
Yes, I would agree with that.
So the kind of experiments you're talking about here, maybe not as commonplace in biology?
I think they're just as important, maybe not as commonplace, because there is a challenge that we have with this generation of students, which is that many students coming to university are very adverse to failure.
Right.
And it is much more comfortable for most of these students to work on a computer and play with electronic data, right? And know that if something doesn't go right, they can just basically erase it and start over. The time investment is small, and there's not a big financial investment. And you put those same students into a wet lab where they have to handle pipettes and moving small amounts of liquids around, and they get frustrated and nervous because they feel like they're going to fail. And with many things in science, especially when you're doing experiments, there's a high rate of failure. But every time you fail, you learn something. So we like to think of it as fail forward, because you're going to gain something from that experiment that will help you in the next round of experimentation that you do. So for me, the most important thing for students that are being trained in my lab is that they have a combination of wet lab experience, and they also have that bioinformatic or data-heavy experience. And that I make them go out and see the organisms.
Yeah.
I just took my lab group out to Virginia a few weeks ago in June, where we went to a couple of shellfish hatcheries to collect larvae. And it was the first time that these two students had actually seen living bivalve larvae under the microscope, and watching scallops spawn in these sea tables and going through that whole collection process. So it's very important that people remember that even when we're working with a small tube of DNA, or we're looking at a giant data set of DNA sequence, it comes from an organism that is interacting with its environment.
What are you working on now? What's next for the work in your lab?
Next, we're really focusing right now on some of the chemosensoring work. So those animals that we brought back from Virginia in June, those will be prepped for sequencing soon. We have multiple larval stages, and we're going to look at how DNA, or how the gene sequence or gene expression, is changing over time in these larvae. The hope is that we're going to be identifying for the first time some of these new chemoreceptors that are occurring in both the scallop and the oyster. And we're going to be using some really interesting modeling methods to figure out what chemicals may be docking into these proteins.
Jeanne Serb, thank you.
Thank you so much.
Voices of Excellence is produced and recorded at the Ohio State University College of Arts and Sciences Marketing and Communications Studio. More information about the podcast and our guests can be found at go.osu.edu/voices. Voices of Excellence is produced by Doug Dangler. I'm Jen Farmer.